Good morning children, I am Sagadisis. Now I am going to teach you the salient features of double helical DNA. As you know, the DNA has double helix structure and it was proposed by two scientists, James Watson and Francis Crick. So, James Watson and Francis Crick in 1953 proposed this double helical or twisted model of DNA based on the X-ray diffraction data given by Maurice Watkins and Rosalind Franklin and Chargaff's rules of base pair. So based on these factors, they were proposed the double helix model of DNA. And among that, the Chargaff's rules of base pairing is very important. Let's see what is Chargaff's rule. Actually, the Chargaff's rule was proposed by Chargaff in 1950. And according to Chargaff, the first point of Chargaff's rule is the nitrogen bases such as arginine is always base pair with thymine, and the cytosine is always base pair with the guanine. So that is the first postulate. In the nitrogen base, arginine base pair with thymine and cytosine, or we can say first we can write the purine. So guanine, arginine and guanine are purines. Base pair with the pyrimidine cytosine. So these two are pyrimidids and these two are purines. So these are the nitrogen bases, purines and pyrimids. So these two purines are base pairing with these two pyrimids. Okay. So arginine is always base pair with the thymine and guanine is always base pair with the cytosine. And the second one is the amount of arginine is equal to thymine, and that of the amount of guanine is equal to cytosine. Okay. The amount of arginine is equal to thymine and the amount of guanine is equal to cytosine. Okay, the amount of arginine is equal to thymine and the amount of guanine is equal to cytosine. Okay, and the third one, next we can see the third point of Chargaff's rule. The arginine always base pair with the thymine with the help of two hydrogen bonds. Okay, two hydrogen bonds. So arginine base pair with the thymine with the help of two hydrogen bonds, and the guanine always pair with the cytosine with the help of three hydrogen bonds. Three hydrogen bonds. So that is the next rule. So arginine base pair with the thymine with the two hydrogen bond and the guanine base pair with the cytosine with the three hydrogen bond. In turn we can say thymine base pair with the arginine with two hydrogen bond, and we can say cytosine base pair with the guanine with the three hydrogen bond. Okay. And the next point, the ratio of arginine to guanine. The ratio of arginine to guanine is, and the ratio of thymine to cytosine is always equal to 1. So the last point is the ratio of arginine to guanine and that of thymine to cytosine is always equal to 1. Then we can write like this. A plus G, C plus T. Or because G plus C is equal to 1. Okay. So, this is the Chargaff's rule. Is it clear? Next we can see the salient features of double helix model of DNA. What are the salient features of double helix structure of DNA proposed by James Watson and Francis Crick? Okay, the first point is the DNA is made up of two polynucleotide chain. Okay, and the second point is in this polynucleotide chain, the nitrogen bases are complementary to each other. So, or we can say the two polynucleotides are complementary to each other. They are complementary to each other. That means the two strands are complementary to each other, or we can say there is a complementary base pairing between two strands. Okay, and the third point is the two strands are anti-parallel. That means there are two strands, they are helically twisted, and the one strand is having polarity. One strand with polarity 5 prime to 3 prime, or we can say 5 dash to 3 dash, and other strand with the second one with the 3 dash 5 dash, or 3 prime 5 prime polarity. That means the two strands are having anti-parallel polarity. Okay, they are anti-parallel. And the fourth one is in the two strands the nitrogen bases are base pairing. That base pairing is adenine is always base pair with the thymine, and cytosine is always base pair with the guanine. Or we can say guanine always base pair with the thymine. Okay. Sorry, cytosine. So here adenine is always base pair with the thymine with the help of 2 hydrogen bond, and guanine base pair with the cytosine with the 3 hydrogen bonds. That I already taught you. And the next point is next salient feature, the diameter. I will show you. So here we can see the nitrogen bases guanine, cytosine, adenine, thymine, etc. So the distance between two base pair is 0.34 nanometer. So here we can see this is one base pair, another base pair, next base pair. So the distance between two base pair is 0.34 nanometer. Distance between two base pair is 0.34 nanometer. And next we can see in a complementary, or we can say in this complete turn 10 base pairs we can see. In a complete turn, here we can see 10 base pairs we can see in a complete turn of this double helix structure. So, in a complete turn, 10 base pairs are there. 10 base pairs are there. Okay. So, these are the main salient features of double helix structure of DNA proposed by James Watson and Francis Crick. Hope you understood everything about the salient features of double helical DNA.